Every cell in a living organism's body has a strand of DNA. That DNA contains all the information necessary to create all of the parts of the body and to run all the processes of the body. Every time a cell is copied, its DNA must be copied as well, so that information is passed on. In this chapter, we are going to study DNA and how it is replicated. This is Chapter 14, DNA, the genetic material. Before we talk about the structure and replication of DNA, I want to talk about how it was discovered. We're going to begin with the work of Frederick Griffith. He was working with two kinds of bacteria, rough and smooth. He injected rats with smooth bacteria and they died. He then injected rats with rough bacteria and they lived. He heated the smooth bacteria and killed it. He then injected this dead bacteria into the rats and they lived. Finally, he mixed the dead smooth bacteria with live rough bacteria and injected it into the rats, and the rats died. He observed that something was transferred from the smooth bacteria into the rough bacteria, and he called this material a transforming principle. He called it this because the bacteria was transformed. This was the initial discovery of DNA. He just didn't know what he had. Our next group of scientists are Avery, McLeod, and McCarthy. They were interested in what the transforming principle was, and so they modified Griffith's experiments. They began by heating the smooth bacteria and killing it. Then they took a portion of it and treated it with the enzyme DNase, which digests DNA. They mixed it with the rough bacteria and injected it into the mice, and the mice lived. Then they took a second portion and treated it with protease, which digests protein. Again, they mixed it with the rough bacteria and injected it into the mice. This time, the mice died. They identified the transforming principle as DNA. They had considered the possibility that the principle was protein, but this experiment confirmed the presence of DNA as that transforming principle. Hershey and Chase began with a bacterial virus. This is a virus that infects bacteria. They chose this virus because it is made with an outer coating of protein and has a core of DNA. They grew the two groups of viruses in a radioactive solution. One solution made the protein coat radioactive, one made the DNA radioactive. They allowed both virus groups to infect bacteria. The bacteria that had been infected by the radioactive DNA virus created more radioactive viruses. The bacteria infected with the radioactive protein did not. This proved that DNA was the source of hereditary material for the cell. Now that the biologists knew what DNA was and what it was capable of, they wanted to study its structure. In the 1920s, a biochemist named P.A. Levine was studying the DNA molecule. He discovered that it had 4 nitrogen bases: adenine, thymine, guanine, and cytosine. In the 1950s, Erwin Chargaff began his work, and he found that the nitrogen bases were always found in similar quantities. He also discovered that adenine always bonds to thymine and cytosine always bonds to guanine. This means that there is an equal proportion of adenine and guanine, called the purines, to cytosine and thymine, called the pyrimidines. Rosalind Franklin used X-rays to determine that the DNA molecule had the shape of a helix or a corkscrew. In 1953, Watson and Crick combined the discoveries of Chargaff and Franklin and others who came before them to identify the structure of DNA, specifically that it has a double helix. DNA is composed of millions of subunits called nucleotides. Each nucleotide is made up of a 5-carbon sugar called deoxyribose. A phosphate group is attached to the 5th carbon. If you look at the strand on the left, you'll see the phosphate. It's a little circle with a P in it, and it's attached to the 5th carbon. It's written as 5 with that little line. That's called 5 prime. A nitrogen base is connected to the 1st carbon, or 1 prime. A hydroxide molecule, or a hydroxyl group, OH, is attached at the 3 prime carbon. Nucleotides are connected at that 3 prime carbon. The hydroxide group above is attached to the phosphate group below, creating the DNA strand. The sugars form the backbone of this strand. When scientists are looking at a DNA strand, they always read it from top to bottom, top being 5 prime and bottom being 3 prime. The nitrogen bases of one side of the helix are attached to the other using hydrogen bonds. In case you missed anything that I said on the previous slide, I'll go over it one more time. DNA is a nucleic acid. Remember, we talked about nucleic acids briefly in Chapter 3 when we talked about macromolecules. It's composed of repeating monomers called nucleotides. Each nucleotide has a 5-carbon sugar called deoxyribose, a phosphate group, remember the functional groups we talked about, that's attached to the 5 prime carbon, and a pair of nitrogen bases, one on each side. Those bases are adenine, thymine, cytosine, and guanine. And then there's a free hydroxyl group, or a hydroxide, OH, that other functional group that I talked about, and it's attached at the 3 prime carbon. And this is just a visual so that you can see all those pieces, parts I just talked about. The backbone of DNA is made up of everything but the nitrogen bases. So that's the sugar, the phosphate, and the hydroxide or hydroxyl group. The two backbones are joined by the nitrogen bases. As I said before, adenine can only join with thymine. This is because they form 2 hydrogen bonds. Guanine can only bond with cytosine because they form 3 hydrogen bonds. This also puts a purine with a pyrimidine. This is called complementary pairing, or adenine and thymine, guanine and cytosine are called complementary base pairs. The entire DNA strand is said to have anti-parallel configuration. This means that one side starts with 5 prime and the other starts with 3 prime. All organisms are composed of DNA strands that are put together in the exact same way. What makes one organism different from another is that nitrogen base sequence. Each nitrogen base sequence creates a different protein. Those proteins are the basis for all of the processes and all of the cells. You put those together, you get different organisms. And this is just a visual of everything that I talked about. This is Watson and Crick's double helix. The DNA molecule is the genetic material that determines the structure and the function of the organism that it's inside. That structure and function is then based on the creation of proteins. And as I said, that sequence of nitrogen bases is what proteins are based on. That DNA molecule, however, can come in a variety of forms depending depending upon what's going on in the cell. When the cell is in interphase, or doing its job, DNA is found in a long, double-stranded fiber called chromatin. Prior to replication, the chromatin will thicken and coil and become a chromosome. In order to keep the DNA from getting tangled up as it compacts, small sections of chromatin are wrapped around groups of 8 histone proteins, which are then attached to a nucleosome core, and the entire thing is called a nucleosome. Being tightly coiled does 2 things. 1, it allows a lot of information to be packed into a tiny space. And 2, it ensures that the chromosomes are properly separated during mitosis and meiosis, or cell division. A single chromosome, just one from mom or one from dad, is called a homolog. A pair of homologs, whether it's one of mom's and one of dad's, or the original and the copy made during replication, is called a homologous pair. After replication, each chromosome is made of 2 identical DNA molecules held together by a protein called a cohesion protein. These 2 chromosomes are now called sister chromatid. Having identical chromatids allows the cell to have equal distribution of genetic material when each cell is created. In other words, you're going to take the original and you're going to copy it, and then you're going to split those cells. You want a copy of the exact same DNA in each cell. The point where the sister chromatid are held together, that cohesion protein, is called the centromere. On both sides of the centromere, on the outsides of the chromatid, are the kinetochores. These are disc-shaped proteins where the spindle fibers will attach during cell division. At the end of the chromosomes are telomeres. They're not shown in this picture. A telomere is just a piece of DNA that doesn't have any genetic information. There are a series of repeating pieces of DNA, like AAAA, CCCC, that protect the chromosome from breaking down while also limiting the number of times that the cell can divide. I'll explain this more in class. In any organism, the chromosomes are made up of 1 chromatid from mom and 1 from dad. This is called diploid. Humans have 46 diploid chromosomes, or 23 pairs. In a gamete, which is an egg or a sperm cell, there's only 1 chromatid. So in the egg, it's only 1 chromatid from mom. In the sperm, it is only 1 chromatid from dad. This is called haploid. If a mistake is made in cell division and you end up with an extra or a missing chromosome, you can end up with extensive damage, or it could even kill the organism. Now it's time to start talking about actual DNA replication. In order for replication to occur, you need 3 things. You need something to copy, a template, which is the original DNA molecule. You need something to do the copying, and this will be enzymes. And you need the materials to do the copying, which are individual nucleotides. DNA replication is called semi-conservative. It's called this because 1 strand remains intact, called the parent strand, during replication. That strand is copied, creating a new double helix. The new DNA strands are called daughter strands. The new DNA, when it's completed, will be made up of 1 original strand and 1 new strand. The process has 3 steps. Initiation, which are all the preparatory steps. Elongation, where the DNA is actually added to each nucleotide one at a time, and termination, all those processes that tie everything together and complete the DNA. I'll start with initiation. Replication begins at a specific site called the origin of replication. The enzyme helicase opens and unwinds the DNA strand, forming a Y-shaped replication fork. As the helicase unwinds the DNA, it forces the double helix in front of it to untwist. A group of enzymes called topoisomerase will keep the DNA from getting knotted as it untwists. Single-strand binding proteins will be attached to each of the uncoiled strands to keep them separate and keep them from snapping back together. The enzyme primase will set up the last portion for DNA replication. It will add a short piece of RNA called RNA primer at the origin of replication. When the RNA primer is set, initiation is over and elongation can begin. Once the RNA primer is in place, the enzyme DNA polymerase can begin elongation or actual replication. It will attach a nucleotide to the RNA primer and begin the complementary strand. DNA polymerase can only add new nucleotides after the RNA primer is added. When you look at the screen and you're looking at the template strands, the blue lines, you'll see 5' at the top and 3' at the bottom on the left, and the opposite on the right. DNA polymerase must add nucleotides toward 5' and toward the replication fork. The strand that's able to do both of these things is called the leading strand. Nucleotides can be continuously added in one long strand because it's doing both of the requirements. The complementary strand on the opposite side builds toward 5' but away from the replication fork. This strand is called the lagging strand. The lagging strand is made in small parts. To begin construction of a new strand on this side, RNA primer must be added to give the new DNA nucleotides something to attach to. The nucleotides are added in groups of 3 called Okazaki fragments. Each time a new fragment is made, the polymerase moves toward 5' but away from the replication fork. Because of this, after each segment is added, the DNA polymerase has to go back to the replication fork, a new RNA primer must be added, and then a new section can be made. Once all of the Okazaki fragments have been added, and the leading strand is all the way at the end, elongation is over. The Okazaki fragments will then be connected using the enzyme DNA ligase. The RNA primers will then be removed and replaced with DNA nucleotides. The binding proteins that keep the strand apart will be removed, and helicase will be removed as well, and the strand will be allowed to twist back up. When that happens, termination is completed and the 2 new DNA strands will have been made. This is another visual to show you how these 2 strands are made, and then this is a summary of how DNA is replicated. The writers of the AP exam love to have students compare things, and one of their favorites is the comparison between prokaryote and eukaryote DNA. When we look at shape, prokaryotes have circular DNA, eukaryotes have linear or double helix DNA. Complexity of prokaryote is simple, no chromosomes. Eukaryotes have chromosomes coiled around histone proteins, making them complex. Size: prokaryote DNA is small with a few number of genes. Eukaryote is large with extensive genetic information. Replication for prokaryotes is simple because they have only one origin of replication, because it's circular. Eukaryotes have complex replication because it can have multiple origins of replication. Gene expression: for prokaryotes, most of the genome is expressed, meaning if the trait is in the genome, you get to see it. For eukaryotes, only a portion of the genome is seen. The organization of DNA and genes into chromosomes gives eukaryotes benefits that prokaryotes don't have. We'll cover a lot of this in mitosis and meiosis and genetics, so if this seems a bit vague, don't be too concerned. I'll also be covering this in detail in class. So first we have genetic variation. It allows for independent assortment between chromosomes. This means that an offspring has an equal possibility of getting a mom chromosome or a dad chromosome. It allows for crossing over. This is the exchange of information between a mom chromosome and a dad chromosome. We have improved genetic stability. This allows genes to be linked and inherited together. It makes the transfer of genetic information more efficient and helps to prevent loss of information because you have a backup chromosome. It allows for increased complexity. This allows more genes to be squished into that small space that is the cell, and it increases the opportunity for evolution. And finally, it allows organisms to be diploid. Being diploid reduces the effect of harmful genes because you have a backup. It also reduces the harmful effects of not getting a gene or not getting a chromosome because you have a backup. And finally, it allows organisms to be heterozygous, which means you have a dominant and a recessive gene. This also increases genetic variation. Mistakes happen during replication. DNA polymerase has the ability to proofread its work and make corrections, but mistakes still happen. These mistakes are called mutations. Mutations that occur during DNA replication of the DNA in an egg or a sperm is the source of natural selection. Mutation is necessary for evolution, but there can't be too many mistakes or the organism will be too flawed to function properly. Another way you can get an error is through a mutagen. A mutagen is any agent that increases the number of mutations above a normal expected level. We have chemicals such as things that we put in our food, we have nuclear radiation, and we have sunlight, which is actually UV radiation. Cells have a variety of ways to repair damaged DNA or to correct errors that were made during replication. DNA repair is vital to the cell, and that's why the cell will spend so much energy on different repair systems. There are two types of repair categories. First is specific repair. This targets a single kind of error in DNA and repairs only that type of damage. The second is non-specific, and this uses a single repair mechanism to fix multiple kinds of errors in DNA. And we will look at an example of each type of repair. Photorepair is a specific repair mechanism to repair damage caused by UV radiation. UV radiation causes thymine bases that are next to each other to covalently bond together instead of bonding to their adenines. In photorepair, the enzyme photolase absorbs light and uses that energy to split the thymine bases and glue them back to the adenines where they belong. Excision repair is a non-specific repair that will correct a variety of different problems. First, the cell recognizes that there is damage to the DNA. An enzyme specific to this repair will attach to the damaged area and a single base on either side. It will cut that damaged area and those bases out and throw them away. DNA polymerase will come in and add the proper nucleotide by matching it to the complementary strand. It'll bring it in and glue it just as it did when it was making DNA. And this repairs the little piece that's missing. This completes Chapter 14, DNA, the genetic material.